Okay, hey, how's everyone doing tonight? What we're going to do is we're going to go through some slides tonight. I'm going to try to slow down here because I really want to talk fast. But we're going to go through some slides tonight, and I want you to take some notes. Most of the slides you're going to have to take notes on. So what I really would like you to do is listen to this presentation first. It's only going to be about 10 minutes. And then take notes afterwards. Don't try to take notes during the slide because I'm going to tell you some things that I don't want you to write down. It's just for your information. Okay? So what we're going to do is we're going to start and we're going to look at the cell. And we discussed in class that the cells are the basic structure. They're the basic structure and function in living things. We looked at how the buildings that are built with bricks, and we related that to what cells are. Cells are the building blocks of life. Okay? How can we see cells? Well, cells are very, very small that we can't really see them with the naked eye. I mean, I can see my skin and I can see, but what I can't see is individual cells down there in my skin. So what we need to do is we need to make a slide with a microscope to see inside of our cells. A microscope is an instrument that makes small objects look larger. And in class, we've been looking at microscopes, and the microscope that you have is a compound microscope, and the compound microscope contains more than one lens. There's also something called a simple microscope, and that contains one lens. So for instance, if I was using a magnifying glass to try to see something with just one lens, that would be a simple microscope. If I'm using two lenses, one at the top and one at the bottom, or more than one, it's going to be a compound microscope. We looked at the microscope and we saw how it worked, and we saw how we needed a light source to go into the little hole on the stage of the microscope. What that does is that lens, or that light, goes into the lenses, and the lenses will focus it on the object. If you look at a lens, if you look at my glasses here, the microscopes are sort of similar to this in where my glasses are a little bit thicker in the middle than they are on the outside, and they're curved. They're not straight. They're not flat. They're curved, and the same type of thing used in eyeglasses. The type of lenses it uses is called a convex lens. Okay. Today in class, I was using the analogy of a curved shape. Now, there's two types of curves. If you look at a spoon, when you're eating your cereal, there's a curve that goes inward this way. Okay? That's called a concave type of curve. It's caving in. If you flip the spoon over and look at yourself like this, or if you flip your spoon over and try to eat food with it, you can't eat it that way because it's not caved in. What we call that shape that rounds out like that is a convex. And if you look over here, right in here, this is convex. So it's thicker here than it is on the sides. Okay? What happens is light goes in, and the light will pass through, and when light hits the eye, the object looks bigger. Okay? We don't really need to know this part in here. You're going to learn that in 9th grade in your physical science class. When we talked about our microscopes, we talked about two things and what must be combined. One was called magnification and the other one is called resolution. With magnification is the ability to make things look larger than they are. Okay? On this picture right here, Here's the naked eye. Over here is 4 times the magnification. We have 6 times the magnification. We have 8 times magnification. We have 10 times the magnification. We have 20 times the magnification. What's happening is each time it magnifies, things will get closer to us. How do light microscopes make things look bigger? Well, the lens or the lenses in a light microscope will magnify the object by bending the light that passes through them. You do not have to write this part down. Okay? This slide is, you can do this again in 9th grade. But right here we have a light bulb and we have the light coming through here. And if you could follow this, and I can't make it any bigger, but as light goes through the lens, it gets bent right there. Okay? And then it gets bent to a point, and then as it goes up the tube, it spreads back out again. And when it spreads back out over here, when it spreads back out, what we have right here is it bending one more time. And then at the top lens, we have magnification. Okay? We talked about each lens will contribute to the magnification of an object on your stage. So if we said the eyepiece that we looked through has a 10 times magnification, and then when we look through the bottom, whichever one that we look at, we have to multiply that number. So the total magnification is equal to the 2 lenses multiplied together. Okay? Over here, if your eyepiece was 10 times, then, and your objective was 100 times, 10 times 100, so it would look like it's 1,000 times bigger. Okay? You need to know this part right here, when 2 lenses are multiplied together. That's magnification. Let's look at resolution. Resolution is the ability to clearly distinguish the individual parts of an object. So when we look at something, is how clear do we see that? Another term of it is for sharpness. And what I keep saying is take a look on your cable box or your satellite box and go to a non-HD channel and look at the picture that's on there. And then, that's if you have an HD TV. And then go to an HD TV channel. The same channel, just keep flipping back and forth. And what you'll see is the resolution is better on the HD TV. Okay? So if your microscope is based on 2 things, magnification and resolution. You want to be able to see it very clear. There's other kinds of microscopes that your book goes through. Electron microscopes. And these use beams of electrons instead of light to examine a specimen. Too technical. Okay, your book goes over a couple of the electron microscopes, but that's about it. That's about the scope of this class, is electron microscopes. We don't really get to use them, so the book doesn't really talk about them. But they've made us see things clearer, and their magnification is even more than the light microscope. Let's take a look at Robert Hooke. Robert Hooke was a scientist. He was one of the first people to observe cells. He made his own microscope. When he looked at the cells, he observed cork, and he found cells in there. And in these little tiny cells, what he said was, hey, these look like they can fit people in there. I'm going to call them a cell. The name stuck. Okay. There's another guy called Leeuwenhoek. He looked at pond water first with his microscope. He saw little things running around in a pond water. He also took scrapings off of his teeth, put it on a slide. Nasty, but guess what? We're probably going to do that. And he saw bacteria that was on his teeth. It was pretty cool back then. I never knew that. What's this film? They had no clue. All this brings up to us when we start looking at the microscope and how helped us look at cells. We had to come up with a theory on what cells are. And there was a bunch of different scientists collaborating with each other. Okay, they came up with an explanation of the relationship between cells and living things. The first one is that all living things are made up of cells. Everything that's living, everything that was living, or everything that used to be alive. You got to listen to me. Everything that was once alive is made up of cells. Has to be. Because by definition, all living things are made of cells. If it was never living, there are no cells to it. Part 2 says the cell is the basic structure and function unit of life. Part 3 was that all cells come from pre-existing cells. Stop. I know what you're saying. Cells come from cells. That's the theory. But it doesn't explain how the first cell came to be. Leave that up for the future scientists of the world. Okay, but we know that the cells will reproduce and form a daughter cell. Okay? Who came up with all these? The first one was the guy named Schleider. He said all plants are made up of cells. He was a botanist. He looked at all plants and he kept on saying, wait a minute, the only thing I see normal about all these plants, the only thing I can really pinpoint is they're all made up of these little walls right here. So he says they're all made up of cells. Another guy named Schwann came up and he looked at cells. He looked at animal cells. He said, wait a minute, all animal cells are also made up of cells. Well, if you have plant and you have animal cells and they're all made up of cells, what else kind of life do you have? I don't know. This Virchow came in and said all cells come from pre-existing cells. So he came in and said, wait, cells will reproduce. There's no such thing as poof, there's a cell. I don't know where it came from, have no clue. It's almost like when we looked at one of those videos with the maggots, and it said about the maggots don't just show up, they have to be there or they have to be on something first for it to happen. That's it that I have tonight. And here's some random facts that I have. The average human being is composed of around 1 trillion individual cells. That's average. If you're big like me, you have more cells. If you're tiny, you have less cells. And if you take a look at this dot on this letter I, it would take as many as 50 cells to cover the area of the dot on the letter. Well, that's it for this part. I'm going to be done with this one now, and you'll just have to wait till you get the next part.